On behalf of Cambridge Health Tech Institute's Global Web Symposia and our sponsor for today's event, Agena Bioscience, I'd like to welcome you all to today's Web Symposium. My name is Allison Proffitt. I'm the Editorial Director of BioIT World, and I'll be your moderator today. So today we are considering sample identification and quality control with Agena Bioscience's MassARRAY system and how the system is being used to screen paired tumor and germline specimens submitted to the Cancer Genome Atlas. Our first presenter today is Erik Zmuda, Technical Director of the Biospecimen Core Resource Molecular Laboratory at Nationwide Children's Hospital in Columbus, Ohio. Dr. Zmuda is an American Board of Medical Genetics and Genomics Fellow in Clinical Molecular Genetics, who is also currently working as a Molecular Genetics Technical Director in the Department of Pathology and Laboratory Medicine at NCH. Welcome, Dr. Zmuda.
Well, thank you for that introduction, Allison, and hello, everyone. My pleasure to share with you today as a user of the MassARRAY system our experience genotyping tumor normal samples submitted for TCGA. So as an outline for the talk on slide 2 here, I'm going to touch on 4 main points for today over the next 20 minutes. And I'll briefly give you an introduction into what our Biospecimen Core resource is and provide the context of our group within the larger project that's known as the Cancer Genome Atlas, or TCGA. I'll then give a brief intro on what the MassARRAY Platform is, for those who are not already familiar with it. And then I'll talk about how we use that platform to perform the genotyping that we did for TCGA, both as an example of working with an existing panel as well as being able to customize a panel over time as your needs evolve. And then lastly, I will move to some of the informatics tools that can and potentially you could say need to be developed when working with large genomic samples and a lot of genotyping data that's being generated from these. So with that, I'll jump into what our BCR is and what TCGA is as a larger project. And so TCGA, if you're not familiar with that, is an NIH-funded consortium of basically an international collection of tissue source providers, so groups around the world that are providing tumors to this project that channel through our BCR as basically a regulator of checking the quality of these samples and the consistency of these samples with their stated histological diagnosis. And then with DNA, RNA extraction from those samples, if everything passes the QC checks that we do, we distribute those analytes out to sequencing centers where exome sequencing is done, as well as genome characterization centers around the country that look at multiple different platforms from low-pass whole genome sequencing, messenger RNA sequencing, microRNA sequencing, DNA methylation, as well as SNP6 copy number variation. There's also a subset of these tumors that are submitted that go for protein characterization as well for proteomics. So once all of the characterization is complete, then the data that has been generated is submitted to a centralized data coordinating center, where that data is first turned over to internally a group of experts, disease experts, that are collected to analyze the results and are tasked with writing a marker paper that provides the genomic signature of that particular tumor type. And it is, TCGA has published many of these papers that are the specific marker papers. And then after that publication is complete or within a particular window of time, the moratorium on the data is released, and then that data is available for public consumption. So in a nutshell, that's the high-level overview of TCGA, and the goal was to characterize approximately 30 of the most common or most lethal adult tumors. And so we'll spend another minute then drilling down here into what the BCR is. And so I mentioned the BCR sits at the front of this pipeline, this rather expensive pipeline of characterization. And so what do we actually do within the BCR? And our next slide then shows more detail about this, this group. And so again, up here we have tissue source sites from around the world are submitting samples for consideration. And there's a clinical data group that does the initial review of those samples to make sure they meet the eligibility requirements for the study. If those samples do, They're brought into our facility where we have different labs that are responsible for different aspects of the pipeline, from a logistics team that coordinates the bringing samples in and distributing them to the different groups, a histology group that prepares slides and stains those slides so that they can be transferred to a virtual microscopy group where images are taken of the slide and sent out electronically for path review to confirm the diagnosis and then the percent tumor nuclei of the specific sample that we receive. If the sample passes that pathology requirement, then the logistics group transfers the tissue to the molecular lab where I oversee. And within that lab, we do DNA, RNA co-isolation and then several points of QC checks of those analytes. If it meets all of our QC requirements, then we'll create aliquots for the characterization that I mentioned, transfer those to our logistics group, and then FedEx will pick up our cryoports so that those analytes can be distributed to the characterization and sequencing centers. So at this point, TCGA has pretty much reached its end, and so we can take a look back at the volume of cases that came through the pipeline. And so I'm showing some numbers here where we ultimately received 34 different tumor types, and within that there was almost 20,000 cases that were submitted to this project. Those were provided by over 150 different tissue suppliers. And there were approximately 60% of those cases passed all of our QC metrics and went on to ship for characterization. For those that didn't pass, they failed for various reasons, and I show there non-genotype related and genotype related because that's essentially the point of this presentation for today. The genotype related disqualifications accounted for 1% of all of the samples or all of the cases that came in. And so I wanted to kind of emphasize at this point the ramification, or let's say the value added of doing this genotyping test in the context of a large throughput project. With every case that we ship out, the price tag for the characterization that's being done is approximately $15,000 per case. And so by the fact that our pipeline was able to catch samples that did not have an appropriately paired tumor and germline normal, which is required for the study, we were able to prevent an additional $4 million from being sent on cases that ultimately would have been discovered as being a genotype mismatch and kicked out of the project in the end. And so with essentially a relatively minimal investment based on the final savings, so we spent approximately $325,000 on capital and reagents to do all of the genotyping for this project, we were able to save $4 million in sequencing costs by not allowing those genotype mismatch cases to ship for characterization. So this Sequenom platform has performed very well in our hands, and that's one number that can kind of highlight the value that it added to our group. So we take just a minute then and we start talking about what is the molecular pipeline, and maybe I'll kind of gloss over the fact that there are multiple points along our QC pipeline where we have tumor and normal extractions, DNA and RNA are extracted from the same piece, and we look at three main things. What's the yield from the extraction? What's the integrity of the analyte that we get back? And does the genotype of the tumor and normal specimen submitted, do they match each other appropriately? And are they unique to all other samples submitted previously? If all of those criteria are met upon the review of QC metrics, the sample status is changed to qualified and it's prepared for distribution. If it fails any of those, we repeat the extraction and do the process again. And so it is here on the QC step where the MassARRAY panel really came in handy. So what is the MassARRAY platform? Just briefly, I've got a few slides here to kind of simplify the workflow and the overview. Essentially, this is a mass spec approach for identifying the sequence variation at specific point mutations. It involves PCR to amplify the region around the SNP of interest. It involves a annealing and extension primer immediately adjacent to the SNP that you're interested in testing, and then extending that extension primer by one base so that it incorporates the specific nucleotide that you're questioning the identity of. And once that nucleotide has been incorporated, you can transfer your PCR reaction, very small amount of liquid, onto this microchip up here using a nanodispenser. And then this chip is what goes into the mass spec, where the data is generated for what the identity of your SNP is. So with the next cartoon, then we can talk a little bit about how we are able to detect the specific sequence at these SNPs. So here I'm showing a single SNP position and PCR primers that are flanking the SNP of interest. And in this example, I'm providing a heterozygous patient where each allele has a different nucleotide. And after PCR, you end up with a population of those two sequences being amplified. In principle, it should be an equal mix of the two alleles. After PCR, you add your extension primer. As I mentioned, it's immediately adjacent to the SNP, and extend that by one base, so that what you're left with in the end is a population of extension primers that have that nucleotide incorporated into it. Once that has been added, then you can resolve the identity of that base by running this on mass spec, where it's exquisitely sensitive to the mass of the extension primer that you put in. So here's one example of what that mass spectrum looks like for one sample. You can see here the mass of an unextended extension primer. So after the reaction, in some cases, not all of the extension primer is used up, and so you can see a little bit of a residual peak here. And then note the large shift in mass as, in this case, the T or the C has been added, and you have a nice gap between the mass of those two options. So essentially, this is, in the raw sense, what the data looks like. And this is for one sample at one allele, or one SNP. And so now if we take that same SNP and we plot this out for a population of samples, the point of this graph here is to give you a flavor for what the data looks like as you accumulate more data points over different patients. And essentially what you have are the masses that represent the two axes. So anything that is homozygous for the low mass of the two options hugs one axis, or the high mass hugs the other axis. These data points represent homozygous patients where there was only amplification for, or let's say extension to one or the other of the alleles, suggesting they're homozygous at that position, or you have an even mix of the two, suggesting that they're heterozygous. So that is one SNP. Now really the power of MassARRAY is in the ability to multiplex many SNPs into a single reaction. And the intention of this figure here is essentially to illustrate that the footprint of the example that I just showed you for one SNP would occupy this relative amount of space on the mass spectrum, and it leaves quite a bit of bandwidth to add in additional SNPs so that you can generate a more robust set of data to test whatever it is that you're interested in. Today we're talking about sample ID, but you can also use this approach to genotype other disease-related markers that are traced through SNPs. So essentially all that is required is some optimization of primer compatibility, as well as increasing the mass of your extension primer so that these little clusters of unextended versus extended masses have enough space between them so that they're not overlapping and creating confusion within what peak is attributed to what extension primer. So with that said, we'll turn back to our project and talk about how we applied this platform to our genotyping needs. I'll reiterate that the goal for our pipeline specifically is to confirm that tumor and normal submitted for each case are genetically matched to each other, and that case has not previously been submitted to the project. And that's a tricky piece because tissues are coming to us from cooperative groups or institutions that are not necessarily knowingly working together, but maybe they have the same patient in their repository. Anyone was eligible to submit tumors to this project, so we needed to identify duplicates based on genotype, not based on the sample identifier that was being provided by an external site. So these are our two goals. We initially started our genotyping using SSTRs, but quickly realized that for the scope of the size of this project that was not sustainable. And that ultimately really only allowed us to perform the tumor normal comparisons. It was very difficult to do the historical matching feature using just SSTRs. So this was back in around 2010 when we were looking for an alternative to SSTR, and I think around the same time, Agena, or I think Sequenome at the time as it was called, was working on developing their genotyping panel. And based on the relationship that existed between our BCR as well as the second BCR at the time, the International Genomics Consortium and Sequenome, there was collaboration between our groups to develop the initial sample ID panel. And this initial panel was a highly multiplex reaction that focused on 45 Sniffs that were selected to achieve the maximal resolution of identity. And for just the simplest sense, the identity resolution power of these panels are extremely sensitive. And down at the bottom here, I've shown you the random match probability based on the first version that we used. And it was essentially the chances that any two genotypes from different people would be the same using this panel was 1 in 5 trillion. So this provided a tremendous amount of resolution, giving us confidence that we could use this panel to accurately detect mismatches as well as historical matches submitted to the project. And I'll just note that these calculations are based on Caucasian genotype frequencies. So with that said, this slide here essentially gives you a flavor of what the genotyping output can look like. It also gives you the specific dbSNP numbers for the SNPs that were included in this panel. And I think a couple things that I wanted to say about this slide was that we initially started using this for DNA, and we found that this panel did work very well for DNA. We had very few failed loci, and it gave us very good ability to resolve cases. But over the course of time, as identity investigations cropped up, we tried testing RNA on this platform, and we found that the RNA worked as well. And what we suspect is that we're able to detect the genomic DNA contamination of RNA within these samples and actually genotype off of that contaminating DNA within our RNA. So obviously with the DNase treatment, that signal goes away, but for the purpose of this figure, I wanted to show that both DNA and RNA can be run on this, and this MassARRAY panel is so sensitive that you can pick up trace amounts of DNA in a solution of otherwise purified RNA. So this version here was the first one that we used, and we processed approximately 53,000 samples on that. Everything was going well for a couple years, and then we heard back from the TCGA program office, and they asked us to customize the panel based on an evolving need of adding sex markers to the panel so that we could confirm that the molecular identity or the molecular sex of the specimens that we received matched the clinical sex of this, the clinical data that was being reported for those cases. And they also asked us to include coding regions of the genome so that when we conducted our identity investigations, if issues arise in the future, we would have the ability to compare our genotyping results to the RNA sequencing results, as that was a relevant issue at the time. So the problem with that was not identifying which SNPs to use for this addition, because Agena had already validated the sex markers, and the BCRs had previously validated the C-SNPs, but those ultimately didn't make it into the first version of the panel. So we knew which SNPs to add, but what we suspected we were going to have a problem with is that we were going to run out of room on our mass spectrum to add more SNPs in. So I mentioned we already had 45 SNPs in the panel. Now we were talking about adding 3 more sex markers, and the program office asked us to consider 15 C-SNPs. So that would have taken our total to 63. We knew through Agena that the recommended max was 40, and we were already exceeding that. So we had a problem that we weren't completely sure how to address. We knew we wanted to be able to compare the genotyping results from any revision of our genotyping panel to our existing database of the 35,000 patients, because that historical match was still important. So we had to decide how to revise the panel. So the way that we approached that was to identify the SNPs that were providing the least amount of information to our historical matching. And the way we looked at that was we said, well, from all of the samples that we ran, what SNPs give us the same genotype at a very high frequency? And we drew a threshold at 60% of the time if we got the same genotype back for all samples at one loci 60% of the time, then that really wasn't as informative for us, and so it won't hurt us that much to remove it from the panel. Conversely, we wanted to pick up more SNPs that had a higher fail rate than the others and remove those because they're failing frequently. They're also not contributing to our historical match very much. So this graph here shows on the y-axis, we've double labeled the frequency as well as the fail rate. So frequency is in blue, greater than 60%, made the cut for being considered for removal, and then the rest of these red arrows represent the SNPs where there was a higher than expected fail rate, or let's say the higher end of the fail rate for all the samples that we ran. Now the next slide, I'm going to show you the final result, and I'm skipping the 3-ish months that we spent optimizing the conversion from our 45 to where we ended with was 50, and it was essentially add in all of them so that we were running 63, and then start to remove the ones that looked like they were either competing with each other or had already been identified as ones we probably wanted to remove so that we could minimize the change. And in the end, we found that we only had to remove 8 of the existing SNPs, so 2 of them because they had too frequent, or they were associated with a highly frequent, gave the same result, several that were failed, and then 1 that we didn't really pick to remove in advance, but it was conflicting with our new SNP, so we had to remove it. And down here are the SNPs that we added in. So this is what our current version looks like. As of the beginning of this year, we have genotyped over 23,000 samples using this panel, and everything is going well. We have been able to identify historical matches just as well as we could with the original panel, and we're very happy with the final result from the optimization. But the optimization of this was key so that we could minimize the number of times samples had to be repeated when loci fail. So with that setup, that's the panel that we're using. And so now I want to transition. If I have any time left, please let me know if I'm running long. What are the informatics needs to support the genotyping approach that we're doing? And I thought it would be maybe interesting for the listeners to see 2 approaches. One is a quick and dirty approach for small projects. This is something that essentially I personally developed out of need based off of one-off side projects that utilize this machine. And then the other is the professional approach, professional informatics support team that has allowed us to run TCGA-style projects. I don't believe that many people have the IT team that we're blessed with here, so that's why I thought it might be useful to show just 2 extremes at the end. Again, the goals of our case, the goals of this remain the same. Tumor normal must match, and they must be unique. So what's a quick and dirty approach? You can use so to make this simple, I guess we'll just say you can do pairwise correlations on the genotype output from the sequenome. And a very simple and fast way to execute those pairwise correlations is using this R program, which is essentially a language and an environment for statistical computing. I'm not an expert in R. I could not write an R script on my own to save my life or anything, but I do know some people who can script R, and I said, I have this problem and I need your help. And they sent me through a link instant message this text here, and I was able to basically parrot this through R, map it to the correct files that I needed it to be mapped to successfully run pairwise correlations using this very powerful environment. But it's very easy. I know nothing about it, and I was able to figure it out in about 10 minutes. So I'm extremely confident that anyone out there can do that. Now, to get to that point, you have to do a little bit of Excel jockeying to get the MassARRAY output file into a compatible format for the R program to run it. And it's just a few steps where you essentially convert the nucleotide calls to a numeric code, and that is a very simple within Excel. It's just find, replace all, T's with 1, C's with 2, etc. As you can see, even the heterozygous you can replace with 12, so that every one of these combinations of nucleotides is unique. Once it's in this numeric format, you can use the pivot table feature within Excel to very easily create this grid where all of your patient samples are across the rows, and all of the SNPs for those samples are across the columns. Once it's in this format, then you can even stop here and basically just eyeball the relationships. You can see if the genotypes are similar or not. I personally would not do that. That would probably be very error-prone and hard on your eyes. But it's in this format, you basically take it and put it into that script that I showed you on the last slide. And what it gives you in the end is this output file that has turned your sample list. So the rows here are now a pairwise comparison. So it's the same set of patients across the rows and the columns, and it's all pairwise correlations of how similar were all of those genotypes to each other. And if you format this table so that each of your samples that were submitted with each other, so in this case it's patient X10 where their metastatic as well as their primary tumor genotyping results were in adjacent rows, you see the correlation of the genotypes is very high for those two samples. Next patient pair, same thing. You can kind of walk across this table looking for the correlations. You get down here, we see this guy's a little bit less of a correlation, and so it allows you to go back into the raw data and look to see if there's an issue with that sample. In this particular instance, it was just a tumor pairing that showed a high degree of loss of heterozygosity between the two, and that affected the pairwise correlation. So I want to emphasize that this visualization is not to make a determination on whether or not something is a mismatch or not. It is simply a way to visualize the data that allows you to quickly hone in on issues. Obviously, if you have a perfect match or a near perfect match in the genotype between any two samples, you have high confidence that they are from the same individual. But where that actual line of mismatch is in this format is not well defined. It's just a way to highlight samples that you should go back and look at a lot more closely. So here, using this approach, then, I wanted to show you an example of what a mismatch looks like. So we've moved to a different set of patients. It's the same format where we're pairwise looking at the correlations. And as we move down, we see, we hit here where now we don't have that nice grid of 4 where the correlation of genotypes is very high. When you go back into the raw data for this one, we actually do see there is a mismatch between these two samples. So this approach was able to identify that mismatch. So that was one of the objectives, was do tumor and normal submitted for a case match each other. The other scenario is, are there any historical matches within the database? Now this quick and dirty approach is much more challenging to do that because the historical match, you're going back to everything you've received previously, and if you've received a lot of samples previously, you really need some other tool to do this. But if your population is a fairly small, manageable cohort, you can use this same table. I'm actually just showing you a zoomed out view of the last two slides where here was the first set, here was the second set that has the mismatch. And then so to find the historical matches, you're looking for high correlation off the center line. So anywhere out here, if there's a sample that has a high correlation with another sample, that would suggest that it's a genotype mismatch. This real set of samples did not have a mismatch, or excuse me, did not have a historical match. But as a control for this process, I introduced a mismatch that is up here. You can't see it, but it says fake duplicate as a positive control for the ability to detect that mismatch and here detect that historical match. And here's what that looks like, is it shows up as a high correlation off the center line. So not to belabor that part too long, that's a quick and dirty approach to use basically a script to do pairwise correlations to look at your genotyping results so that it is not a manual review of the raw data in all cases. So for TCGA, we used an informatics team to generate quite a few features that allowed us to achieve our genotyping goals. And it's, again, the same goals. The only thing I'll emphasize differently here is that we needed this thing to be scalable for high throughput. In fact, the peak of what our processing was within TCGA was 10 genotyping plates per week, where we had technicians that were reviewing the results and genotyping approximately 400 to 500 cases per week. So within that, the workflow that was developed was very nice, actually. So we integrated the pre-MassARRAY steps of even generating a sequenome input file from our limbs to a post-MassARRAY processing pipeline where we ultimately just had a drag-and-drop network directory that we could take the sequenome output files to, and it would go through a scoring application and a data warehouse, where various steps were performed in that data warehouse, including the automated genotype comparisons. Now, maybe just to emphasize the scale that we achieved with this TCGA project, as of the 58,000 samples that we have in our genotype database right now, every time we drop another plate into that scoring algorithm, there are 1.6 billion comparisons that are performed through this approach. So this size of a project really does require a sophisticated tool to be developed for it to run efficiently without error. I've shown here some of the rules that we applied for the automated genotype comparisons, and I maybe won't spend too much time on that here because I'm going to show you an example of the output file, because the reporting of this is important. So again, if our goal is to achieve scale and we want to be able to sign out 400 to 500 cases per week, we need there to be an efficient report for interpreting the results. And so the next slide, I'll show you what that result looks like, and it's essentially data is grouped by plate, the patients are grouped together, and we went to great pains to format this report in a way that made it impossible to ignore genotype issues. So here's an example of the report that we use. We start over here. I mentioned that the samples are grouped by plate, and so we would pull this report based on the plate identifier that we put into the Dropbox. And then within that report, when it pulls up, the samples are grouped by patient. And then within any, so patient A, patient B, patient D, within each patient, you get all of the samples that have been genotyped for that patient, regardless of what plate they were run on, as well as any patient that exists in the database that has greater than a certain degree of similarity based on its genotype to the patient that you're currently looking at. So in this case here, patient B, we've got the blood and the tumor genotype aligning to each other, but it's also brought in patient C, because patient C actually has very high, if not perfect, genotypic match to patient A. So that was how we were able to detect or visualize historical matches within the database, and then by aligning the tumor and the germline with each other here, we can visualize the mismatches. Some of the other things that we incorporated into this, and then also to illustrate the red bolding here, is that patient should not have been pulled into this block, and so the logic knew that was the case, and so it lit those cells up as red. None of this was done manually, other than I edited the patient IDs to make it generic and not release any TCGA specific information. We have some additional QC data. It gives a quantitative summary of the homology between the samples using the blood as the best normal, and then how similar are any other sample within that patient to the best normal. It summarizes for you how many loci failed, and we had an internal quality metric of there had to be fewer than 10 failed loci for us to accept the genotype result. If there was greater than 10, that was unusual based on historical performance, so we repeated the sample. And then we also created this easy score, which is a very lowbrow quantification of the type of mismatch. So if you have homozygous germline SNP where you gain an allele in the tumor, that is not expected based on what we know about tumor biology. The opposite is possible, so that would be a phenotype known as loss of heterozygosity. So here you've got a heterozygous down to a homozygous. That's well-known cancer biology. This is unexpected, so we penalize that. Easy score value of 1, and then anytime there were heterozygous mismatches, we put that as an easy score value of 100, and then you sum those across the row so that you can just immediately know this patient has 5 homozygous mismatches and 3 heterozygous mismatches. And it gives you some extent of the degree of difference between the genotype of the tumor and the normal, or any two patients relative, any two samples relative to the blood normal. There's a way to get rid of that flag. The next piece over here is the sex markers that are included in the genotyping panel are summarized here, and this is illustrative of the two outcomes. Our report, because we wanted to know the molecular sex so that we could QC it against the clinical data that was being submitted for each of these samples. So the report brings in the clinical data and reports that this clinical data sex is male. The molecular data supports that it's male. So the only thing that changes here in the comparison between these two is that it goes green if they match, or if the logic doesn't. So we wrote the rules to say what a male was by the genotype. If the rules were broken, then it doesn't change the call for the clinical data, but it just lights up that cell as red so that you look and say, well, that particular sample has a mismatch with the clinical data and the molecular and sex data. So with that, maybe I'll just run through the examples here and also say we also brought in all of the raw data so that we could visually inspect the actual calls for each of the loci. So in this first example is the perfect case. There's no genotype issues. We have a nice match between the germline normal and the tumor, and TCGA did do whole genome amplification, and we genotyped those as well, and there were never issues detecting the genotype on WGA. The next example is a scenario where a duplicate patient was submitted. So we see patient B and patient C are showing up. They all have a perfect genotype match to each other. You inspect the raw data and it checks out. The molecular sex matches the clinical sex, and so that would flag us to do an investigation into how we received two samples that have the same identity. And in almost every case, it only required pulling the path report, and upon inspection of the path report, you could clearly see they were from the same patient. The next example is a swap tumor where patient D and E have a mismatch between the germline and the tumor. So there's only 34% between the blood and the tumor here, and an EASI score of 53. So that's flag 1. And then the feature here is flag 2 is we also have a historical match where patient E's tumor is matching the blood. And so that query to historical match also allows you to unlock these reciprocal swaps where it's not just that the tumor and the normal submitted were from the wrong patient, it's somewhere along the way, and we never pass judgment on if it was within the BCR or prior to the BCR, something was mislabeled. So either the blood or the tumor from these two patients had been mislabeled. This genotyping approach and this particular algorithm of comparing all the results against each other allows you to find that reciprocal match. And then the last example, I think, is just to illustrate the genotype is a nice match, but the clinical data for sex doesn't match the molecular data for sex, and so then that requires a follow-up investigation. So with that, I'll just summarize and wrap up. The MassARRAY platform is absolutely a genotyping workhorse. We have been able to generate robust multiplex assays for anywhere between 30 to 50 Sniffs per well without really any trouble at all. I'd like to say that MassARRAY is not just for identity testing. We've had a lot of success using it for HPV typing as one example. As far as the TCGA projects, HPV is a relevant viral infection for cancer development, and so we were able to use that to type the particular type of HPV that was present in different tumors, as well as various assortment of disease-associated SNP screening. There are low-tech and high-tech analysis options for the data that comes out of it, and based on our experience, the first thing we do, or the most rigorous thing we do, is optimize any new assay very early on, because the repeats accumulate very quickly when you start running a lot of samples through this machine in bulk. So early optimization allows for fewer repeats later. With that, I'd like to acknowledge all of the people who have contributed to the development of the MassARRAY and worked on the TCGA project, both here at the Nationwide Children's Hospital BCR as well as IGC BCR. So I'm just showing a few of the PIs here, program managers, the technicians who work in the lab, as well as the informatics staff, and then similar, the lab staff and the program directors from IGC. My thanks to Hema from Agena, is one of the most effective technical support people I've ever had the pleasure of working with. He has solved so many problems for us. I couldn't even summarize it. He is an amazing asset. And then also my thanks to Katie Hoadley from UNC for providing or sharing with me her expertise with R coding. So with that,
All right, thank you.
Back over.
Yeah, so everyone, if you have questions, please go ahead and be sending those in as we go, and we will get Darryl's slides queued up Darryl Arwen is the Director of Applications and Technology at Agena Bioscience. He manages the scientific operations in the Asia-Pacific region, including customer support, genetic services, marketing, and product development activities. Darryl, if you are ready, we are ready for you.
Thanks, Alison, and thank you to Dr. Zmuda for showing us a very clear utility case study of using a custom genotyping identity panel on the MassARRAY. So I like to think of an experiment like I think of a healthy meal. And when we're thinking about an experiment, we often think about the hero. When we think of a meal, we often think about the hero of the dish. That might be a nice, big, juicy steak, piece of salmon, something else. When we think of an experiment, we also think of the hero of the dish, the DNA analysis on the cohort. But just like a meal, we need on the side some healthy greens and vegetables to keep ourselves healthy on the inside. We need sample ID and sample QC to keep our experimental pipeline healthy. And Dr. Zmuda showed us the reality that is even in their very expert hands, 1% of samples were mixed up and were able to be identified by a genotyping panel. In fact, studies have shown that anywhere from 2 to 20% of samples and cell lines in various different biobanks across the world are actually mislabeled or contaminated. And that has actually caused the retraction of a number of high-profile papers because the scientists were not characterizing the samples that they thought they were characterizing. So several years ago, the Sniffs for ID consortium showed that Sniffs could be used as a method of sample identification, and that generally anywhere from 30 to 50 high minor allele frequency Sniffs is sufficient to identify any 2 DNA samples, whether they be human specimens or human-derived cell lines. So Sniffs are a well-accepted method of identity characterization. Now, on the other side, what's the importance of doing sample quality assessment? Now, one of the challenges that we see when we have poor sample quality is not always that the sample fails, but the sample can work, but we have a problem called stochastic noise. And this is a major source of experimental error, especially in poor quality clinical samples. And that is where low DNA input copy number can lead to false negative results. So let's look at this population here. And in this population, if we take a statistically sufficient sampling, we have 15% red shirts. However, if we take only a small sampling from this population, and let's just take this middle one here, if we take this as our sample, then what we actually see here is an upward bias of our red shirts. So this moves up to 60% red shirts. And if we take this down here, which is another small sample of the population, we can see we've got 0% red shirts. Now, rather than being red shirts, let's change that over to somatic mutations. And in this instance here, we have an upward bias in the frequency of somatic mutations because we've taken a small number of DNA samples which was not representative of the original population. And down the bottom here, of course, this is more the serious problem in somatic mutation screening. In this unrepresentative sampling of the population, we have 0% mutation frequency. Not because the technology had an error, just simply because that is what we put into our reaction. So we all need to respect that clinical samples are limited in size and they're frequently degraded. But the reality is, if you want to detect a 5% mutation frequency, then you need to put in 300 intact DNA template molecules. That is, 15 of those input DNA template will be a mutant strand. If you go below that, you're going to move into this area of sampling error, possibly upward bias, or the very dangerous downwards bias in your mutation frequency. And we've seen this before. Here is an example of when we first validated our OncoFocus panel. And with our OncoFocus panel is detailed mutation screening in core genes involved in oncology: BRAF, KRAS, NRAS, KIT, and EGFR. And in this, we ran a set of 68 samples, FFPEs, and cell lines, all that had known mutation signatures. So we had 64 known mutations. And we ran this brand new panel across these samples, and we only detected 58 of the 64 known mutations. But when we ran our sample ID and QC panel across those samples, we found some very interesting things. Well, first of all, 3 of the 6 missed mutations were at less than 10% frequency. And when we ran the sample ID panel and looked at the QC markers in that panel, we identified that all 6 of these samples where we'd missed the mutation had less than 100 intact DNA template copies going into the reaction. Basically, this was the example of sampling error, where we hadn't put a relevant set of DNA template into the reactions, and we'd frequently missed low frequency mutations due to sampling error. When we came back and increased the DNA input such that we did have a representative set of DNA template in the reactions, all 6 mutations were easily detected. Now, there's another challenge in clinical samples, which are generally formalin-fixed paraffin embedded, and it's not just that they're fragmented and low copy number, but also the formalin causes some artifactual changes in the DNA sequence. Now, this study was published by the group at the Peter Mac, Hung Do, and Alex Dobrovic just 2 years ago. And what they're looking at here is this challenge of formalin artifacts when you put in a small amount of DNA template into your reaction. Now, the formalin causes C to T changes. And here, when they're putting a small, they've got some very old FFPE blocks here, and when they're only putting a low amount, so 5 nanograms of FFPE into the reaction, we see this increased amount of C to T and the corresponding, if you're looking at the opposite strand, G to A changes. If the amount of input DNA is increased up to 25 nanograms, and that is, instead of only having small amounts, a larger amount, all of these samples come back as wild type. And this is an example of stochastic noise because causing formalin artifacts to be seen as true mutations. These formalin-induced C to T changes happen randomly across the DNA sequence at a generally low level. However, if you only put in, say, 10 molecules of DNA template to amplify, and one of those molecules happens to have a formalin artifact, well, that will be seen as a 10% frequency mutation. Whereas if you put in an increased amount, because of the low frequency of these artifacts, you won't observe them as a mutation. So I come back to the point that to detect a 5% mutation, you need to ensure that you're putting in at least 300 intact DNA template into the reaction. So now on to our iPlex Pro Sample ID panel. Our iPlex Pro Sample ID panel is a single reaction consisting of 44 SNPs for identification as well as 3 gender markers for identification, and it has 5 copy number control assays. So the combination of 44 SNPs and 3 gender markers, the 44 SNPs selected are a high minor allele frequency across all populations. 44 SNPs and 3 gender markers give us the power of discrimination of 10 to the minus 18. So again, an extremely high power of discrimination. And seeing there are only just over 10 to the 9 people on the Earth, 10 to the 18 certainly gives us sufficient power to identify individuals. So we get this high confidence sample matching. Now, we don't really need 10 to the minus 18, although it's useful to have for sample-to-sample matching. But as Dr. Zmuda pointed out before, when you're trying to do tumour to normal pair matching, there is a challenge that occurs. In tumours, we have loss of heterozygosity and copy number gain. So any genotype, any marker that is a heterozygous genotype in the germline could be any genotype in the tumour. That is, if you're GA in the germline, you could have lost the G and become homozygous A in the tumour. You could have lost the A and be homozygous G, or you could be heterozygous. I think everyone will recognise that if you could be any genotype in your tumour, there is very little power in that matching. Now, with 44 SNPs, high minor allele frequency, 22 of these will be heterozygous on average in any individual's germline. Now, we exclude those because they give us no power for matching. So that leaves us with 22 homozygous SNPs, and one of the things that does not occur in tumours is a gain of an allele generally, particularly with the markers that we're using here. A gain of allele is not expected, so we have 22 homozygote assays which we can use for high confidence matching, still giving us 10 to the 9 power of discrimination. The copy number control assays give us accurate quality assessment, telling us the absolute number of amplifiable DNA template copies that are in that sample. And the entire panel is coupled with a completely automated matching database for storage and matching software with summary reports and other outputs that I will showcase in a moment. So we have a variety of applications for this sample ID panel. Authentication of cell lines. Cell lines are being shared across the world frequently, and they're frequently not authenticated. Chain of custody events. I'm going to show you an example of where we've used this panel to barcode a germline DNA sample, barcode the library prior to next generation sequencing, and check that barcode in the sequencing data. And of course, I've mentioned before the disease and normal pairing and the determination of absolute amplifiable copy number. So let's have a look at what the matching process is. So the matching process, a perfect match is a score of 10 when we're looking at non-tumour versus non-tumour, so germline versus germline. So a perfect score is a 10, and a match is considered anywhere from 0 to 10. The way the software works is we penalise mismatches. So we penalise mismatches based on the severity of the mismatch. So that is heterozygote to homozygote change is less severe than a homozygote to homozygote change, and so on. And a mismatch is a score of less than 0. With the non-tumour versus tumour pairing, now we generally have only half of the markers being informative because we rule out all of our heterozygous germline markers. So a match is 0, again penalising mismatches, but not penalising mismatches that are heterozygote to homozygote changes now. And any non-zero value is a mismatch. So the clearance criteria for this panel, and this is user modifiable, but these are the standards that we have set in the software as the default. So any samples that have less than 30 successful SNP genotype calls will fail. The software will fail the sample and not use it to do matching, and that's because we're starting to lose power to make an accurate match or mismatch identification because of the reduced number of markers. And samples that have less than 500 amplifiable DNA template copies will fail. Now, we recommend putting in 10 to 20 nanograms per reaction. 500 copies represents around about 1 nanogram of intact DNA. So we can use this absolute copy number to determine the number of intact DNA template molecules in these challenging clinical samples, formalin, fixed, paraffin embedded samples. and to determine whether the sample is suitable for robust short amplicon technologies such as the MassARRAY and panels like OncoCarta, or whether there are higher copy numbers with longer intact template that is required for technologies such as next generation sequencing. So here is an example of one of the reports that comes out. Here is on the screen in front of you is our sample report. You get one of these per sample. Sample name, when it was run, what position in a plate, the number of amplifiable copies. You can see this is a very good quality sample with around about 17,000 amplifiable copies. Our 3 sex markers giving us a genotype of female. Here we have the SNP barcode, so we've got all 44 SNPs being called and the barcode for the sample. And down here we found in our comparisons one unexpected match in that AS263 has unexpectedly matched AS2_2, and the green lights being all of the genotypes that have matched here. Moving on, here's an example of an unexpected mismatch. So these 2 samples having the same sample name and same sample identifier. And where you're able to set the software for a sample identifier that determines a section of the name that is used for identification. And that is, if you have replicates, you can have the same sample identifier with _1, 2, 3, 4, 5, or _T for tumor, so on and so forth. And here, these 2 samples should have matched. The software was expecting them to match. We have a score of -156. Now, remembering anything less than 0, we assign as a mismatch. And here we have lined up the different genotypes that have mismatched. Heterozygous mismatches in yellow, homozygous mismatches in red. So clearly these are actually different individuals. Now, moving on, very similar to what Erik just told us about. Our customers also asked us if we could design a panel that had coding SNPs and SNPs from gene regions in it, because all of the sample ID SNPs were in intronic regions and non-coding regions. So we designed a panel which we call our Exome ID Assays by Agena panel, 42 SNPs, 3 XY paralogs, and 5 DNA QC assays. The inclusion criteria for the SNPs were that they needed to be located in gene exons and UTR regions such that they're captured by exome capture kits. The SNPs need to have high minor allele frequency. We also chose SNPs that were present on several of the cytogenetics arrays, so we could do exome sequencing matching as well as cytogenetic array matching, so copy number array matching. And a secondary inclusion criteria there that it was on the Illumina chip as a lesser requirement. In addition, so you'll recognize that we have 5 DNA QC assays in there, just like the sample ID panel. With the sample ID panel, all of those assays are at 100 base pairs in size. Whereas with the sample QC panel, because we are barcoding for technologies that require longer amplicons, we have put these QC assays at 100, 200, 300, 400, and 500 base pairs in length. So now we are able to determine the number of amplifiable copies at each size of amplification. We've got varied gene loci, but all the genes that are selected for these QC assays are stable, or at least as stable as you can get somatically, never seen double deleted, which allows us to do that QC for downstream applications that require longer templates. And we can also generate a degradation curve, and that is the number of molecules versus the size. And let me show you some examples of those. So here we have some examples of some FFPE samples, and you recognize FFPE samples are heavily degraded. All of these samples had 10 nanograms of total nucleic acid input into the reaction. And what we can see here on the x-axis, we have the size of the amplicon, 100 through to 500 base pairs. On the y-axis, we have the number, the absolute number of amplifiable DNA template at each size. So you can see here at 100 base pairs, we've got just over 4,000 intact DNA template. At 200 bases, that drops to just under 2,000. 300 bases, it's at 700, and we have no amplifiable template at 400 and 500 base pairs. So clearly with this sample, if you're trying to do clinical Sanger sequencing up in the realms of 400 to 500 base pairs, it's not going to be successful. You really need to keep it into the shorter 1 to 300 base pair amplicon size. Well, let's move all the way down to the bottom right-hand corner here. Another FFPE sample where at 100 base pairs we've got 250 copies, 200 base pairs we've got 70 copies, and 300, 400, and 500 base pairs we have nothing at all. Well, here absolutely we are going to need to keep it to a short, robust, short amplicon robust technology such as the MassARRAY. Even targeted next generation sequencing is going to move into the realms of sampling noise and formalin artifacts. So 2 case studies here. First one here is from the Ontario Institute of Cancer Research, and they're using Agena's sample ID plus assays by Agena panel compared to traditional STR profiling for validating archival FFPE samples. We've heard the challenges that occur with misidentification of cell lines. In fact, Dr. Zmuda told us that he saved $3.9 million for the TCGA by detecting his 1% of mislabeled samples. So we know the challenges here, but the challenge with FFPE is that STRs that are generally several hundred bases, amplicon size, are usually are often not amplifiable off degraded FFPE samples. And the results of this study, it proved the utility of Agena's short amplicon sample ID plus panel in generating genotype profiles from FFPE samples and matching them for downstream next generation sequencing applications, but also the sample ID being short amplicon SNP genotypes provided more definitive tumour and normal pair matching for these FFPE samples than the corresponding STR profiles, because it is more robust on degraded samples. In case study number 2, we're using our Exome ID panel for exome sequencing workflow QC. So in this particular case study, we had 20 patients and all up 88 samples so we had a germline blood DNA sample from each patient, primary tumour DNA sample from each patient, and various different metastatic tumour DNA samples. All of these had also been converted into exome libraries. So the challenge that we were presented with is to make sure that all of these biological samples matched, and also all of the exome libraries that were generated matched, because this is a reasonably complex workflow that has the potential for sample mix-ups. So we've mentioned before that tumour DNAs have the chance of less loss of heterozygosity and copy number amplification compared to a normal germline sample, when in fact, next generation sequencing libraries have exactly the same possibility. Biased amplification, which can be observed as loss of heterozygosity or copy number amplification. So effectively every sample, whether it be a tumour or a library, is being paired back to our germline sample using our tumour versus normal matching algorithm. Well, let's go right to the results, because I've explained the study. So we had 88 samples from 20 unique patients. All tumour DNA samples matched the corresponding normal DNA sample. So the tumours, the barcoded or labelled tumours and normal DNA were all matching and from the same individuals. However, we observed 8 unexpected mismatches, including 2 normal DNA libraries that did not match the normal DNA, and 6 primary or metastatic tumour libraries that did not match the normal DNA. So we found significant sample mix-ups in the process of exome capture and library preparation workflow, which were able to be deconvoluted before putting on the next generation sequencer. And here are here's the report. So here's the report of the unexpected mismatches. So we had 88 samples, around about 1,500 comparisons, took 5 seconds to run. And you can see our 8 identified mismatches here. First one here is a normal library, not matching a normal DNA at a score of -30, and so on and so forth. So very clear detection of these unexpected mismatches that occurred in this cohort. Okay, so what do you get from Agena's iPlex Pro sample and Exome ID and QC panels? Well, they're single reaction panels allowing high throughput, low cost sample identification and tracking, whether it be sample matching, disease and tumor matching, or next generation sequencing and array workflow control. Comprehensive coverage with 42 to 44 SNPs and 3 gender ID markers, giving us 10 to the -18 power of identification, and more than enough power to do tumor and normal pair matching. Our copy number assays are what are incredibly unique. No other ID panel available enumerates the number of amplifiable DNA copies at the same time, basically combining your OD, pick-a-green, QC requirements with your barcoding and QA requirements of ensuring sample authenticity. And all of this comes with a completely automated summary reports and sample reports with a database that will allow you to do local and historical matching. So with that, I thank those attendees for your time and hand the ball back to Alison.
Darryl, thank you very much.